So let's share the lesson. Okay, lesson 3. Okay, lesson 3. Let's see if I can make it even smaller or bigger. Okay, you guys ready? All right. Yeah. So the objective of this lesson is to understand how cells, it doesn't matter what kind of cell, it could be animal cells, plant cells, bacterial cells, fungi, which are mushrooms, and then protists, which is the fifth kingdom of all the species that exist on Earth. And all of these species are made of cells, and inside cells we have DNA. The DNA contains the genes, okay? Now, these genes that are carried by the DNA, they are regulated by enzymes, proteins, inside the cell. Now, depending on the type of cell, depending on the type of species, like for example, bacteria versus plants versus animal versus mushrooms versus protistans, the gene expressions are different, okay? They're different. The more complex the species is, like humans, the more complex the control, okay? So let's start and talk about how gene regulation occurs in bacteria, because they are the easiest. Now, bacteria are also known as prokaryotes, okay? Prokaryotes. Now, I always like to circle the word so that you can see. Let me use green, okay? Right here, this is the word, prokaryotes. This is another word for all bacteria. So if you use the word bacteria, it means all the different types of bacteria. You can use the word microorganisms, that means bacteria. Or you can use the word prokaryotes, which are bacteria. These are, prokaryotes refer to a certain type of cell. So bacteria, these are all bacteria, okay? Some are rod-shaped, some are round with clusters, some are other colors, some have spikes, things like that. But there's a variety of different types of prokaryotes, okay? Now, prokaryotes, they don't have a lot of DNA. So that means they don't have a lot of genes compared to you and I, right? You and I, we have a lot more genes. And so cells, bacteria cells, or just cells in general, they want to conserve, okay? They want to conserve. The key word is right here, conserve. Conserve means you don't want to waste. You don't want to waste what? Energy, number one, and you don't want to waste resources. You don't want to waste food. You don't want to waste supplies that are needed to make a gene, to make a protein, right? The purpose of gene is to make a protein, some kind of protein. And so cells in general, they want to conserve energy and resources for other stuff that is necessary, that's needed at that moment. So what our cells do is they regulate gene expression. Now, bacteria, they respond to changes in the environment much faster than you and I, because they're simple. And because they have less genes, it's the process is a lot easier for them in controlling their expression. Now, one location where gene expression is controlled is transcription. Transcription is when DNA, if you remember, DNA, the D doesn't look like a D, but I try my best, okay? DNA, we have DNA. Cells have DNA. Bacteria have DNA. When DNA are transcribed into RNA, that is transcription. Transcription is where a lot of the control by bacteria occurs. So from RNA, you get your protein, P for protein, okay? P for protein, pro. I'm gonna write the word pro. Pro for protein, okay? So transcription changes DNA into RNA. Translation changes RNA into protein. The protein goes to your body and it repairs, fix, update, maintain your body, your cells, to keep you alive and happy. So where does gene regulation occur? It occurs right here. It occurs right here, this X. It occurs where DNA are transcribed into RNA. So it controls transcription of these cells and other cells too. Now, let's move on to how the breakdown works. So inside cells, okay, inside bacteria cells, the DNA is divided up into regions. Now, depending on the number of genes a bacterial cell has, it has segments on the DNA. Now, all the DNA that a cell has does not mean that everything represents a gene. Certain parts of the DNA of the bacteria or you and I, they're junk DNA. Junk DNA means DNA that doesn't represent anything. Then there are regions where they represent genes, okay? And depending on how many genes a cell, a species has, it's embedded in the DNA. Now, every single gene that a species has starts with a region called a promoter. Let me change color. Called the promoter. The promoter is in front of the gene. So if this line represents the gene, the promoter is right here in front of it. And then the DNA continues, of course. So the promoter is right here. This is the G. G for gene. Okay, G for gene. And this is the P. P for promoter. The promoter is always in front of the gene. So promoter is the start region of a gene. Now, it's also the region where enzymes of your cells, like for example, RNA polymerase, which carries out transcription, they bind to the promoter. That's how they know. That's how they know where the genes are. They look for a promoter. So every time they find a promoter, which is a certain sequence, okay, it has a certain order, AUG, okay, it has a certain DNA sequence. Then the cell knows that after the promoter is the gene, and then at the end it would be stop. S for stop, okay? S for stop. And then there would be junk DNA, and there would be another promoter. And it keeps on continuing. Promoter, and then gene, and then stop. It keeps on doing something like that. Now, so next to the promoter, there's another region. I'm gonna use another color. There's another region. This region right here. Next to the promoter. This is called the operator. O for operator, okay? The operator is kind of like the on and off switch, on and off. So the promoter is the start. It's the identity. It's the kind of like the start of a gene. The operator is the region of the DNA where it tells the enzyme, hey, you get to carry out transcription and translation, or you don't get to. If it's off, you don't get to. If it's on, you get to. Move ahead. So it's kind of like the light signal. Green, red. Okay, green means go. Make your protein. Red means stop. You can't make any protein. So that's what the operator does. Now, there's a third name. It's called the operon. Okay? The operon, people, is the entire region. So every gene, which includes the promoter, the operator, the gene, and the stop, right? That is one, this is one operon, okay? That is one operon. So let's say if a cell has 10 genes, so hence, if a cell has 10 genes, it would have 10 operons. So an operon is a region of your DNA where there is a promoter, an operator, a gene, and a stop. So if humans have 10,000 genes, that means how many operons do we have? 10,000. Because every single gene starts with a promoter, and then an operator, the gene, and then stop. And then what's after the stop? Nothing. It's DNA, but it's nothing. It's just junk DNA. It just doesn't represent anything. And then when it gets to another gene, what happens? Promoter, operator, gene, stop. Okay, guys, we good? Does that make sense? So that is an operon, okay? That is an operon. Now, in bacteria, I'm going to use an example of bacteria. Now, in bacteria, there's an operon called lac operon, okay? This is highly studied, okay? Lac operon is one of the first operon that was researched by scientists going back decades ago. And because of lac operon, they understand how it works, how gene expression works. So, the lac operon is an operon found in bacteria. And the list lac operon, which is also a gene because it's an operon, it produces the protein lactose. Okay, right here, lactose. So lactoperon, lac comes from lactose, okay? Lactoperon is a gene that produces lactose, okay? And in what bacteria? E. coli. E. coli is is a common bacteria that is highly studied. Why E. coli? You hear it all the time. It's because it's present everywhere. It's in your body, people. It's in your stomach. It's in your intestines. It's in your. It's around your eyes. It's on your skin. It's all over the place. E. coli is one of the most common bacteria that you would find. Now, so E. coli has an operon, one among many, lactoperon, which produces lactose. When E. coli um, interacts with, let's say, for them to eat, let's say they get a certain food type, okay? Now, let's say you take lactose, right? Lactose, which is a type of sugar, by the way, found in milk. So if you take E. coli and you put it in lactose, okay? What happened is E. coli cannot eat lactose. The reason they cannot eat the sugar is because lactose is a sugar that's too complex for them. So they're not able to eat the lactose. So instead, lactoperon produces a protein that's going to break down lactose into two types of sugar called galactose and glucose. Okay. Once it's broken down to galactose and glucose, E. coli are able to eat the glucose and galactose, and they're happy. They grow. That's how it works. So I'll do it again. Lactoperon is a gene. It's a gene that produces an enzyme that breaks down lactose into galactose and glucose. Because E. coli cannot eat lactose, but they can eat galactose and glucose. So we did a test on this petri dish, and this area does not have see, I can't even see. Yeah, this area does not have lactose negative. Lactose negative means there's no food, okay? There's no sugar. So you don't see bacteria growth here because there's no food, right? There's no lactose, there's no food. Bacteria is gonna die. I mean, you put them here on this petri dish, you incubate it, it's still gonna die. Now, on this side, you see growth of bacteria, E. coli in this case. Now, why does it grow? Because there's lactose. So they grow. So how did these bacteria break down lactose? Because we know they can't eat lactose. It's because the E. coli have the ability to break down lactose into galactose and glucose. So they feed off this sugar. That's why there's so many of them on here. Yeah, guys, are we good? Are we good? Okay. All right. So we know a lot. Here's a little diagram for those of you who still didn't see my point. So here you have double-stranded DNA, right? This is your DNA. And if you look at it, I'm going to change color. So here, from here to all the way over here, this is stop, okay? This right here is an operon. So here's your promoter, here's your operator, and here's your genes. Sometimes when an operon could have multiple genes, it could have up to 3 genes. In this case, there are 3 genes. You see what I mean? It could have 1 gene, it could have 2 genes, it could have 3 genes. Now, why all 3 genes in one operon? It's because all of these 3 genes are related to each other. Their job is the same. They have the same function. That's why they have to be together. But other operons, there's only how many genes? 1. Sometimes 2, sometimes 3, sometimes 4. But usually the multiple gene operons, they work together, okay? They work together on the same function, something like that. And so you see how here the enzyme RNA polymerase attaches to the promoter because it knows that the promoter is the start of every single gene. And then the operator said, go for it, make the protein, right? The operator said, you red light or green light, right? Red light or green light. But the operator said, you can do it, go for it. And so RNA polymerase would carry out transcription to make the to make the message, and then this is the RNA. That's DNA. This is RNA. It makes one-stranded RNA, and then the RNA through translation will make the proteins. This is a chain of amino acid because of translation. See, these three proteins are going to, or enzymes, they are going to go somewhere and perform the exact same job together because they're found in the same apparatus. Now, to give you a little better view, okay, here's another example. Here's E. coli lac operon acts like a switch, okay? So here, lactose operon is off when lactose is not present. Lac operon is on when lactose is present. So how does it work? So let me change the color. Okay, so here is your DNA, right? Here's your DNA. This is gene number 1. It's so hard to see. Let me change it to okay, this is gene number 1, gene number 2, gene number 3, and then right here is your stop, right? Stop. Stop codon, remember that. So this is your DNA. Now, in front of the gene, there's an operator. It's right here, operator. In front of the operator, there's a promoter. Now, why do you need a promoter and operator? Because your enzymes will not know when the genes will start and end. It knows when it ends because of the stop, but it doesn't know when the gene's going to start, so it needs a promoter. The promoter is the kind of like, hey, gene is starting, come here, come here. RNA polymerase, come on over. The operator will tell the RNA polymerase, hey, do you get to carry out transcription or no transcription? So here in this example, you don't have lactose. Let's say there's without lactose, right? There's no sugar, lactose, in the food supply. So what happened is you don't need these genes because there's no lactose. And so what happened is there's another enzyme called repressor. Repressor is an enzyme that binds to the operator. If it binds to the operator, it prevents the enzyme RNA polymerase from carrying out transcription. So this, nothing happens. This one, nothing is made because the repressor binds to the operator, nothing happens, nothing. There is no protein because you don't have lactose. But what if there was lactose, right? What if you took the bacteria and you put it in lactose? Okay, you put it in a petri dish or a beaker containing lactose. Then what happened is lactose is going to there'll be a protein and it's going to suppress the repressor. The repressor cannot bind to the operator, cannot. And then RNA polymerase would bind to the promoter first and it would carry out transcription to make the protein because now there's lactose. In the presence of lactose, the operator is turned on. The repressor is stopped because it's green light now. So, based on this example, you and I have thousands and thousands of genes. The same cell that makes up my skin is the same cell that makes up my bone. The same cell that in my brain, my brain cell, is the same cell for my blood. Now, all those cells, they have the exact same DNA. However, the cell for my blood only makes blood. The cell for my brain only makes brain cells. The cell for my skin only makes skin cells. The cell for my bone only makes bone cells. The reason is the operator turns off and on. If that cell is located in my skin, if it's destined to make skin cells only, all the genes for brain, blood, bone are off. The only genes that are on would be my skin. Guys, does that make sense? Now, when you're young, all your cells are highly effective. You have full of hair, you have lots of hair, your skin is nice and moist, right? Moist, and there's elasticity to your skin. Now, if you look at your grandparents' skin, old people's skin, you notice how it's on the dry side, it's pale, okay? Because you lose that as you age. When you're young, okay. If you have a young baby, look at the baby's skin. It's very nice and soft. You know what I mean? Nice. There is no mole. There is no freckles unless it's baby's born with it. there is no imperfections on the baby's skin because the baby's cells are highly efficient. But as you get older, some of these control regulation, they don't listen to you anymore. So what do? They give you age spots. Remember the age spot I had on my nose? I had it zapped. I had an age spot right here. My wife zapped me last night. I'll show you another one. It's right here. Okay? So I have an age spot right here because I guess the sun, when I run, and it just develops randomly. And so I told my wife, hey, can you zap me? So she put on some numbing cream, and then she used a cauterizing tool, and then she burns it. So she kills the top, maybe 1 or 2 layers of the cell. Because your skin has 5 layers, right? 5 layers of cell. So I told her to burn maybe 2, 3 layers of the cell. And so that's why it forms a scab. And then there's 2 more layers left. So my skin is still protected, but there's a scab. It's dead at the top. So what happened is after 30 days, it takes 28 to 30 days for your skin to completely recycle. So what happened is in 30 days, you'll see me with this for about, if the scab's gonna fall out in 4 days, 5 days. But the skin, the dark spot would go away, hopefully, in 1 month, because the skin sheds, it sheds. And as it sheds, I hope that the cells that did not listen are dead. I hope I kill those cells. You guys got the idea? By burning the cells, I hope I kill them. Now, what if the dark spot comes back? What if it comes back? What it means is the cells that are misbehaving, they are deeper. I didn't do a good job. I need to burn deeper so that I can reach that cell. So I might have to do it 2 times. Okay, does that make sense? It's kind of like freckles. When you get blemishes on your face. Now you're not born with this stuff. Okay, let's say you're not born with. These are not birthmarks. These are age spots that you get because of exposure to the sun, chemicals, smoking, things like that. You get blemishes. You can't erase those. Laser. They're very expensive. So instead of you can use laser, but I use a tool. It's like a needle, and I burn. The needle heats up, and I zap it, and I kill the cells. Okay, guys? Yeah, all right. I hope that makes sense. So it makes sense to everyone. All right, so let's move on to the next idea. Now, so far I've talked about bacteria, how they regulate. Now, if you compare bacteria cells to humans, to plants, to animals, other mammals, to parameciums, okay, this is a paramecium, which is a protist. Now, if you compare prokaryotes, which are bacteria, to eukaryotes, which are plants, animals, fungi, protists, it's not the same. Eukaryotes are much more complex than bacteria. And so the gene regulation has to be more complex. Got it? It has to be more complex because we are more complex than bacteria. And so are there extra steps that's involved in eukaryotes? Yes, a few extra. That's it. Now, so here is an illustration of how it works, okay? So here is your DNA, okay? Your DNA, my DNA, okay? This is your DNA, my DNA. Now, this strand, imagine it's long and it's spiral, double helix, right? Now, this is the gene. The gene's right here. What is right here? This is the S for stop, right? That's where the stop. Okay? That's where the stop is. Because the end of the gene is always stop. Now, how do we know it's a stop? Codons, right? UAA, UAG, UGA. Those are 3 stop codons. Guys, good? Okay? Now, in front of the gene, there's a region called the promoter. Remember the promoter? There it is. The promoter is always in front of the gene. Because that's the only way for your enzymes to know if where the gene starts. So wherever there's a promoter, there's a gene after. There has to be a gene, and then there was always a stop. Now, how do you know it's what a promoter looks like? This is the sequence of the promoter: TATA or TATAAA. These are the 2 sequence of the promoter. So if the promoter is here, The sequence is TATA. It's always the same. Now, the promoter, we can also call it the tata box. Tata, T-A-T-A, tata box. Now, because the sequence is always T-A-T-A. Now, if I look at, let's say, a bear DNA, is the promoter of a bear versus and a monkey, my chihuahua, the pig, the birds, are they all the same? The answer is yes, they're all the same. So I'm not talking about just about humans, okay? I'm talking about all eukaryotes. They have the exact same tata box, which is the promoter. They're all the same. We have the same sequence. The heck? How does that happen, right? Now, so there's a promoter region. Now, there's another region called the enhancer. It's right here, the enhancer. Now, the enhancer is a little bit further above. It's still on the DNA. It's right here. The enhancer is in front of the promoter. And then there's the gene, and then there's the stop. Okay? Now, for this gene to carry out transcription, for this gene to make a protein, what happened is you need several things. Number one, you need RNA polymerase, which is the enzyme that carries out transcription. You also need what is called transcription factors, which are proteins, enzymes that carries out transcription. Now, once these enzymes, RNA polymerase, these transcription factors bind to the DNA, they have to touch. They have to touch. So for them to touch each other, they have to bend the DNA. You see how this piece of DNA is bending? Instead of straight, it bends right here. Because this piece of DNA, this number one, and number two, and number three, they all have to touch each other. They have to touch. They have to touch. They have to touch. So if this number one and number two are the only two touching and not number three, nothing happens. All three, 1, 2, and 3, they have to touch. They have to touch each other. So for them to touch each other, it has to create this bend. You see this bend here? It has to create this bend, this loop. 30 years ago when they were studying this, scientists saw this loop. And they're like, what the heck is going on with the DNA? How come we have so many loops? They're all over the place, these loops. So then as they examined, they studied, they found that every time there's a loop, there's transcription. Every time there's a loop, that means your gene, some gene is being produced. Guys, does that make sense? So to show evidence, visual evidence, so that you can see. So these scientists, what they did is they took the DNA and they put it under an electron microscope. And they saw inside the nucleus, inside the cytoplasm, there are loops. There are loops everywhere. There are loops. So they found out that they're enzymes. So these enzymes, let me clear up, it's getting so busy, okay? So this enzyme, this one, and this one, they all have to touch each other. So something happens inside the enhancer, it has to touch the tata box. This enzyme has to be touching the RNA polymerase. When that happens, transcription is allowed, and it makes this gene, which becomes a protein. Now, this process is a lot more complex than bacteria, a lot more complex, because it requires extra work. Now, so because of that, when you look at eukaryotes, which are these cells, right? These cells. You don't see mutations. You don't see all of a sudden the bear has an extra eye right here. The bear has an extra eye. You know what I mean? Humans, we don't have an extra arm. This guy does not have an extra arm on his shoulder. You see what I mean? Humans, animals, plants a little bit more often because plants have slightly less regulation, but animals, we have very precise control. So therefore, our mutational rate is very low. Not to say it doesn't happen. It does happen, but it's very low because the complex process of gene regulation is much higher than bacteria, than plants. All right? Now, same with protistans. They are the same. They're highly regulated. So, overview. So here is your nucleus, and this is your cell in the cytoplasm, okay? And so transcription is when it makes, let me change the color, green, okay. Transcription is when it makes a messenger RNA using the double-stranded DNA. That's with the help of the RNA polymerase. So regulation occurs here, so that's step one, okay? That's one. And then once transcription is done, that messenger RNA, it gets a cap and a poly A tail. Then it goes into the cytoplasm, where translation takes place. So transcription occurs inside the nucleus, and then translation occurs in the cytoplasm. Here is your ribosomal RNA. This is the transfer RNA is not shown here, but you can see the protein, polypeptide, which is protein, being developed. And the red represents your messenger RNA. This is your messenger RNA, okay? It's being produced. Gene regulation. Now, but there's more. Now, in eukaryotes, because we have so many different types of cells. You see, I have muscle cells, I have blood cells, I have brain cells, I have cardiac cells, I have liver cells, I have intestinal cells. All of these cells, which are called body cells, okay? They're all called body cells because they make up our body. Body cells are different from sex cells. These body cells are also known as somatic cells. Somatic means body cells. Now, these cells are all come from stem cells, okay? All of these cells that we have that make up you and I, it comes from, its origin are stem cells. Now, what's a stem cell? A stem cell is the original cell. The original cell is the sperm and the egg. It's really the egg cell. The egg, and then the sperm enters, delivers the 23 chromosomes, and then it begins to divide, and then it becomes a small stem cell. That is the original cell of all living things. And then these cells, these stem cells, they will become muscle. Some of these will become blood. Some of these will become heart. Some of these will become intestines and liver. Now, all of these cells, are they all the same? Yes. All of these cells have the exact same 46 chromosomes. They all have 46. They all have 46 chromosomes. Now, how come they don't look the same? Because we call this differentiation. That means that inside the nucleus, before transcription, these genes are turned off. Certain genes are turned off. Certain genes are turned on. So therefore, what happened is, if let's say you are this cell, let's say for example, let's say your skin. I don't have any skin here, but let's say you're a muscle. Okay, muscle, right? This cell will destined become muscle. It will not produce a protein for your blood, your nerve, your brain cells, or your heart. It will only produce proteins for your muscle. That's it. So therefore, it becomes a muscle cell. So I wrote here, I said, keratin is an important protein in skin cells, but is not produced in blood cell. Now, does the blood cell contain keratin genes? Yes, but it is off. Got it? It's off. It's turned off. So, in essence, can you grow an ear right on your back? Can you grow an ear on your shoulder? The answer is yes. I can grow an ear right here. I can grow an ear right here. I can grow an arm right here. But it doesn't, because the cells that are here have all the genes for my ear turned off. Who controls it? The master cell. This cell. These cells, guys. These cells. These cells are the master cells. They tell these cells, before they become what they are, they tell them, hey, you need to become a brain cell, and that's it. You cannot produce any protein or anything else, only proteins for your brain, and that's it. Okay, guys, we good? All right, so in the future, if you guys ever, you get older and something doesn't grow right, let's say on your skin, right? Usually you have some kind of mutation. Sometimes that off, it doesn't stay off, it turns on. That's a big problem. Cancer, skin cancer, guys, skin cancer, okay? We call that sometimes a tumor, okay, tumor. Now, so in 1995, this good-looking man here, his name is Lewis, okay, E. Lewis. Now, he is a biologist, a chemist, many things. So he's actually, all of these guys are all teachers. They teach at the college or something like that. And He was the first to prove that there's gene control. There is a gene regulation inside cells. And the specimen that he used are called fruit flies. This guy, this fruit fly. So using fruit flies, Lewis was able to identify that inside cells, inside my cells, your cells, inside our body cells, there's a group of cells that control the regulation of all the other cells. He calls them homeotic cells. Lewis calls it homeotic cells. These cells control what you will become. All right? So these homeotic cells, they are called the master, they control the master genes. And they tell cells to turn off something or turn on something. Off or on. Now, one of the things that people who have cancer, there's many different types of cancer, and a lot of people get cancer. Their homeotic genes are out of control. So when your homeotic genes, and there's many different types of these genes, when they're out of control, you develop a tumor and becomes cancer. That's what happens. So for me, this spot right here, do I think that some gene lost control? Yes. The gene did not behave. I didn't have this spot when I was 20-something, 30-something, 40-something. I started getting it maybe about 3 years ago. See what I mean? I started it 3 years ago. How? Why? It's because the gene, it lost control. And so these homeotic genes, he also calls it homeobox genes, which are the same type of genes that controls activation of other genes. Now, he has another name for them. He calls them Hox genes, okay? So he has several names, but in fruit flies, he calls them Hox genes. And so from his study of fruit flies, Lewis was able to predict what type of cell will produce the eyes, the abdomen, the antenna, the wings, the legs of the Drosophila. He was able to predict. That's how good he got. So when he looked at the embryo, here's the embryo of the fruit fly, he can say that this part of the fruit fly will become the head. This part of the fruit fly will become the abdomen. This part of the fruit fly will become the tail. You see my point? By looking at the genes, and he calls it Hox genes, okay? Hox genes. Now, so after his research of Drosophila, which is the fruit fly, he started testing other species, and he found the exact same thing. We all have Hox genes. We all have homeotic genes, okay? Now, during his research, he also discovered something new. He calls them epigenetics. Epigenetic is another form of regulation inside our DNA. And what it does is it takes chemicals and it tags different parts of your DNA to tell your DNA, do not make this gene. So it'd be something like this. So here's your DNA, right? Here's your DNA, and there's a gene right here. You see this right here? CH3. If this CH3 is called methyl group, okay? This is called methyl group. If this methyl group is attached to that portion of the gene, that gene is not expressed. Got it? So when you are born, before you were born, when you were only a ball of stem cell, Hox genes have already programmed which cells will become your eyes, your brain. Then it also adds epigenetic chemicals to your DNA, and it tells your DNA, hey, do not express these genes. Keep it off for the whole life of the organism. You got it? Guys? Yes. I hope that makes sense. Yes. All right. On YouTube, guys, there is a young man in China. And, in China, there's a lot of people. So there's just more samples to look at. There's a boy, his name is, people call him, it's kind of sad, they call him Two-Face Chang. And he was on a documentary about genetic problems, genetic misregulation. And if you're curious, Go on YouTube and type in Two-Face Chang, and you'll see that he has a face here and he has a face in the back. He has two-face. Two-face. It's creepy. Two-face. Now, do both faces work? No. Only one face works. The one on the back, it doesn't work. It doesn't work, but he has two faces. Now, what the heck went wrong with him? There's a lot of dysfunction in his homeotic genes. I'm sure there's a lot of mutations. There's translocation, all sorts of mutations for him. So he is an example of, a prime example of homeotic genes that's out of control. That's went rogue. Good. Are we good? I hope that makes sense. I hope that makes sense. All right, guys. It's 12 o'clock. You can log out of Zoom. Take a break first, and then work on some of your questions, your notes later. All right?
My loneliness